the link over there. The guidelines themselves are available at the link above. They will be updated as we go. In terms of the House rules, you are unable to speak, unfortunately, in this, but if you would like to ask a question, we have dedicated 15 minutes at the end for a Q&A. If you'd like to ask a question, please do type it in the chat, and then I will monitor them and we'll pick out some of them to answer in the question. Please do try and limit it to questions rather than comments so that we can make the most use of the time that we have to address many of these issues. And so without further ado, we're going to move on to the first of the two talks. Trudy Lin is going to give the first talk. She's a National Department of Health official with the Essential Drugs Program, and she coordinates and supports the review of the NDOH primary healthcare treatment, standard treatment guidelines and essential medicines list. So if those of you who are working in the front lines, like many of us, with the yellow and various colored essential medicines list books, Trudy is the boss of all of us, and make sure that we do a good job in those. She also facilitates the process for these rapid reviews in the management of COVID-19, which informs the management of the guidelines. So I'm going to leave it there, Trudy. You're welcome to share your screen when you are ready, and you can take it from there. Thanks all.
Thank you very much, Jeremy. Appreciate it. So I'm just going to share my screen. I'm not too sure. The pinging is. Can you hear the pinging in the background?
Not no. For me. So no, I think you're okay.
Okay, fine. Thank you very much. Sorry, Jeremy. I'm just trying to share my screen.
No problem. So Trudy's going to start when the screen loads up. Trudy's going to talk us through for a brief, just 10-minute presentation on the discussion really around the process of these reviews, how they're done, how the evidence is synthesized, and where it all feeds out into. And then we'll take it from there. And then the Professor Parrish, who will be the second speaker, will be talking mostly about ivermectin and specifically the evidence around that. Thanks.
Thank you. Are you able to see my screen?
Not at the moment.
Still not. Oh, this is weird. Okay, we'll try again.
It was there.
Yeah, we could see it. There's still a lot of people in the lobby that we're allowing in, so just to highlight that.
All right, so everybody can see that, yes? Can people see my screen? Can I go ahead?
Yes, there's lots of thumbs up for you, Trudy.
Sorry, I'm on presenter mode. Appreciate it. So good afternoon all. So as Jeremy indicated, I'm going to talk about the NEMLC rapid evidence review process. And it's also described in the South African Medical Journal article, which is the reference is provided at the end of the presentation. Not happening. Okay. So this is a living map, which is an indication of how rapidly evidence for COVID-19 evolves. I accessed this map this morning at 8:30 this morning. And as you can see, there's 17,274 articles that have been identified, of which there are 1,157 systematic reviews, 15,630 primary studies, and of which 648 have reported data. Out of interest, since the 20th of July 2021, which is roughly about 10 days ago, there have been 325 more primary studies and 38 more RCTs that were reporting data. So as you can see, there's an overwhelming amount of evidence that needs to be critically reviewed and in an ongoing fashion. So the initial South African COVID-19 response included that South Africa urgently needed to develop guidelines. This would provide guidance for clinical care, as well as inform medicine procurement. But fortunately, there was infrastructure in place, as Jeremy mentioned, in the form of the National Essential Medicine List Committee. And they mandated for the review and updating of the SEGs and the EML, which I'm sure everybody is aware of. The EML, or the Essential Medicine List, satisfies the priority healthcare needs of the population, while the SDG is an implementation tool to implement the essential medicine list. A typical review cycle is roughly 2 to 3 years. However, on the 19th of Yes, and funny. Apologies. Okay, it's the terms of reference, the protocol template, a generic template for rapid reviews, as well as templates for the PICO and rapid review report. These are all accessible on the National Department of Health website. So the evidence review process was adapted to be a rapid, very rapid virtual process. So why rapid reviews? Obviously, quick decisions were needed without compromising the evidence review process. And this was needed urgently to guide clinical practice and procurement, as mentioned earlier on. As you can see on the right-hand side, systematic reviews take essentially years to complete, whilst rapid reviews, under a lot of pressure and duress, weeks to months. So this is basically summarizing the steps in conducting a rapid review. It's all described in the COVID-19 subcommittee's terms of reference, which is published on the website, as well as that SMJ article I mentioned earlier. So the first thing is you have a research question that is received either from the Minister of Health, the National Department of Health, external stakeholders, the Ministerial Advisory Committee, the MAC, the Clinical Guidelines Committee through Horizon Scanning, or through receipt of an evidence alert, whether it be a journal, etc. The subcommittee then prioritizes these questions, and a review team is identified to do the evidence synthesis in accordance with the protocol, which is also published on the National Department of Health website, as I mentioned earlier on. Your PICO question is finalized, and the review team then is required to submit an initial report within 7 to 21 days of PICO ratification. That's quite a lot of pressure. And the subcommittee then reviews the report, and after iterative draft versions, the report is finalized, and the recommendation is then submitted to senior management within the National Department of Health, shared with NEMLC, MAC, and the Guideline Committee, and obviously disseminated for public consumption. Obviously, emergent evidence occurs, so this needs to be continuously monitored, and horizon scanning occurs continuously, and you have the whole process starting all over again. So the first rapid review was published on the 31st of March 2020, and to date there's been 47 reports, and one is currently underway. So the key principle of this process, very important, is transparency. We try and publish everything as much as possible on the National Department of Health website for public consumption, and that is obviously contributes to good governance, which also includes the normal NEMLC governance processes, which is declaring conflicts of interest, maintaining confidentiality, etc. Bias is avoided. Good questions need to be asked in the required PICO format. And decision making is done through a consensus process using an evidence to decision framework, which I'll talk about in a short while. And the delivery time, committee members really try and meet that. And obviously, there's continuous updates as evidence evolved, as I mentioned earlier on. So this is essentially what a rapid review looks like, which is all accessible on the link which I've provided. You have your date, and at the date, it actually tells you whether it's an update or not, your question, as well as your key findings, and then the type of recommendation, the rationale for the recommendation, the certainty of evidence, as well as the review indicator. If you see brackets over there at the bottom, you see a referral to the evidence to decision framework, which is Appendix 4 in this particular report. Which actually facilitates decision making. So the evidence decision framework is a systematic framework that informs decision making to ensure that all aspects are considered. So there's 3 domains essentially. There's evidence, value judgments, and resource use. So it's not only purely on evidence or alone, or on resource use alone. It's got to be considered holistically, and this is what the committee deliberates on very often. Sometimes it takes quite a long time in a meeting to do so. So here's the example for ivermectin, which I'm going to run through very quickly because Prof. Parrish is going to talk to this in detail. The committee assessed the quality of evidence to be very low certainty evidence. For the evidence of benefit, evidence of harms, and whether the benefits outweigh the harms, there was associated uncertainty. Regarding value judgments, feasibility, can the recommendation be implemented? We all know that this is an unregistered medicine, so Section 21 access poses challenges. Regarding values, preferences, and acceptability, we all know that there's no local survey data, but interest groups do support use of ivermectin. And then there's the effect on equity and human rights, and there definitely will be inequitable access of this particular agent because of the Section 21 approval process. Regarding resource use, there's unfortunately no information. So with the rapid emergence of evidence, there's an unfortunate expectation now for the subcommittee to update or review every time new evidence is published. This is really inefficient use of scarce resources. So the committee developed a framework to guide the update of a review. And this is also included in the committee's terms of reference on the website. And the framework is actually dependent on the current decision and the type and strength of the new evidence. This is a summary of recommendations. Thus far, you can see most of them are negative, except for the use of corticosteroids amongst patients requiring oxygen supplementation. 3 agents are currently on the watch and wait list, pending outcomes from large RCTs, preferably in peer review format. So these are the challenges in the process. The preliminary evidence is often observational and often not confirmed by studies of good methodological quality. Recommendations are often required urgently, and this is sometimes before peer-reviewed publications are available and decisions need to be made on preprints. We all know about the retraction of preprints recently from journals. Evidence accumulates and evolves rapidly, so updates are often required. That's a strain on scarce resources. We have limited local data, as I mentioned earlier. International collaboration is key. So one can collaborate with global bodies using global evidence, but this still needs to be adapted to local contexts. Tools does assist, such as Covidence, Revman, and Grade Pro, and development or adaptation processes where you adapt other international guidelines may possibly increase efficiency. But this subcommittee is so dedicated and oftentimes one step ahead of the international guideline panels. So that does provide a challenge. And then there are tight timelines and competing demands on the resources. There's also a possible need to register on Prospero registry going forward, and that requires resources. So I'd like to acknowledge the NEMLC Therapeutic Guidelines Subcommittee for COVID-19, Hero, specifically Jackie Mia for her contributions, the Grade Essay Network reviewers, and as well as other reviewers that have contributed to reports, Cochrane Essay, and the Better Health Program of South Africa. So this is the subcommittee, and they are dedicated to the health system strengthening with regards to COVID-19. They're hardworking experts, and many are frontline clinicians, very dedicated. Most often meetings are held late Friday afternoons when most are preparing for their weekend. Some useful websites and some references. Thank you, Jeremy. Over to you.
Great, thank you very much, Trudy, and thank you very much for giving us such a nice overview of the process behind it. Professor Parrish, you can start sharing your screen if you'd like. Well, I'll introduce you. So Professor Parrish is a current chairperson of the National Essential Medicines List Committee. He's previously been the chairperson of the Adult Hospital Level Essential Medicines List Committee, and also previous member of the Tertiary and Quaternary EML Committee. He has worked in the Eastern Cape for many years at Walter Sisulu University, and again, spending most of his time currently managing COVID patients as the third wave hits the Eastern Cape pretty hard at the moment where he is. He's a physician and former counsellor of the College of Physicians. He is an evidence-based medicine guru. I hope he won't mind me calling him that. He's who I look to if I have any questions about this issue, and we are very excited to hear him talk about the current topic. So yeah, take it away, Andy.
Maybe I need some guidance to do the share screen again. Desktop window, and then window, and then
Trudy, are you able to are you able to unshare?
Maybe that's the problem.
I've unshared, thanks.
Okay. I've got just nothing in my window. That's interesting because I had it there. It's up and about. It's open.
You have to take control of something. Sorry.
Yeah. Sorry, I'm just...
No, it's no problem. So while we wait, I apologize again for those in the lobby. We had an enormous demand on this, which is causing huge amounts of administrative strain to let everyone in all at once. But thank you very much. We do appreciate that. It looks like we may have some progress. It's gone.
Did you see anything now?
Would you like me to share?
I think I can't understand why it's not sharing. I mean, it just doesn't show up in the window, but otherwise, yours showed up in the window, Trudy. You went to share and then choose what to share.
Yes, you must close all your other windows. I think that also will help.
Oh, not the Teams window, though. I haven't got anything else open.
Okay.
I think. Let me just check. Here we go. Oh, that's interesting. We close that up.
I think it gives everybody else an opportunity to join in.
We may have to use your version of it again because I just don't see it. I mean, I've got the thing open and I'm not getting it. It says it is sharing at the moment.
Okay, I'm going to give control to.
Is it sharing now? Hang on a second.
Okay, I've put the presentation.
No, I had it was there. It was there, Trudy. And
yeah.
then it disappeared.
I think it's because I shared. Would you like me to? Let me try and share.
Sorry,
Okay.
I had it there, Trudy. It was gone again. There we go. Is that your version or mine?
It's my version.
Okay. All right. Well, we'll have to work with that then. That's fine then. Welcome, everybody. Apologies for that. I'm not sure why it behaved yesterday, but not today. So if you can just make that full screen, Trudy. Thanks. Okay, so we just want to talk a bit about how we got to where we are in South Africa with ivermectin, which is really a interesting space because it seems to be that most of the sort of professional organizations and opinion leaders have come out on one side in terms of this agent, but there's also a groundswell of enthusiasm for using it, which is often said to be based on good quality or on evidence. And so what I want to do is just run through those things. Next slide, Trudy. How the studies came about and how we look at evidence. And I'm talking about EBM and doctors in general, not specifically about ivermectin. And then we'll have a quick peep at the ivermectin data, and then look at where this all lands up together. Next. So basically, the issue really is that we started out in a strange place as doctors. In 200 years ago, we actually didn't have much, and we had nothing in the way of evidence other than what we saw with our own eyes. And it was only in the last century that people started to get interested in clinician scientists. In other words, people who actually tried to put some numbers and thought behind actually what they were handing out as treatment. And it's also become interesting that part of our professional responsibility is to be custodians of care, to try and make sure that the care we're offering is standardized across all the clinicians. And that gives us a professional responsibility also in how we read and use the literature. Next one, Trudy. So this is really what's happened even just in quite come from the that era, but certainly the era of reading journals in paper and having indexes in paper to the more electronic versions. And the problem really is that as that really reflects an enormous surge in the amount of evidence that there is to get through. And that makes it not only onerous on individual clinicians, but near impossible for any individual to be on top of everything, even within their own field, which makes the idea of groups of people coming together and summarizing it for the good of the medical community very appealing, and something that's led to the development of reviews and guidelines. Next slide. I just want to run very briefly through this. This is the stuff we're all supposed to know, and we spend a lot of time talking about. And it's just really what it means in terms of trial quality. So allocation concealment means there's no chance to select who gets what. You can't choose which envelope of randomized care to give to which patient by holding it up to a light, something like that. Randomization itself is this idea of balancing group characteristics. So two different groups have identical properties, and whatever you're testing is the only thing that differs between the two groups. And then blinding obviously is about neither the people caring for patients nor the people reporting on outcomes being able to know what group people belong to, and so not being able to influence what's reported. Appropriate comparators is important, and we'll come back to that. If your controls shouldn't be getting something which could actually make them get either better or worse. So active controls where the patients get pharmaceutically active ingredients, is problematic in some studies. Patients often get lost to follow-up, and you still need to be counted because you might have been lost for a reason. For instance, if you got really sick and decided to pull out, that's important information. And then again, analysis by intention to treat, I'll cover that with an example later. You have to report before you start what you plan to do, and then actually do it. And the same applies to data dredging. When you change a pre-stated protocol, simply because the results weren't what you wanted and something better comes along which you think you could report. The big question really from the stuff which I think most people learned in med school is there empiric evidence that actually makes a difference, paying attention to these issues of trial quality. Next one, Trudy. So here's, I'm going to run through three very briefly of some evidence behind this. This first one comes from critical care, and it was a meta-analysis of meta-analyses, which basically looked at how these different properties of trials differed between large and small trials. And the bottom line of it, you don't need to go through it in detail, is that small trials were more likely to have problems with the quality components than larger trials. Next one. They then put all this together and said, well, overall, what sort of effect does it have on our belief in a reported endpoint? And the bottom line is there in the little circle at the bottom. This is a ratio of odds ratios. And before your heads get too muddled by that, don't worry about it. It's really just saying that you need to reduce your belief in the size of an effect if the trials are small. It's likely when bigger trials come out that will shrink back towards the null. Next. And here's another example of same sort of thing. Again, I'm not going to spend time on it. Osteoarthritis trials, they called it a meta-epidemiological study, but it's essentially the same thing, shrinking back towards null. The bottom line there, an adjustment of the differences in effect size. Next one. And here's an example from the psychological literature. I've included this not because I'm pretending that our work is essentially like this, but simply because the graph is so pretty. That's a plot, a lightning plot, showing what happens to effect sizes as time goes on. Some of them get better and some of them get worse in terms of benefit. And another example of that is on the right, which is a publication, but essentially showing effect sizes drifting down by about 50% in that particular discipline. This does seem to be to some extent discipline specific, but it is important that small trials seem to give you, for whatever reason, and we'll come back to that, larger effect sizes. Next. So I'm just putting this up not because people want to be inflamed by reading Ioannidis' paper. This is from 2005. It's one of the most published or most quoted papers in the medical literature, actually, and people got very upset about it. But the bottom line really is, moving on to the next slide, please. From that, they really, he did some sort of calculations, and we can talk about what that might mean. But the bottom line again is that if you had underpowered, poorly performed phase 1 or 2 randomized controlled trials, and their prior probability, in other words, the chance that what they were trying to prove was actually true, was low, in other words, 20% or so, then even when they were completed and reported positive findings, you still were less than 50% sure that that was true. And the same applies to even a meta-analysis. So if the studies are inconclusive or small, you change probability, and that will depend to a large extent on how biased they are. So even having a meta-analysis doesn't prove that you've got a belief that's likely to be stable. It might change quite a lot. Next. So the first pit stop is just a summary then. Small trials lead to unreliable measures of effect. Publication bias is one of the reasons for that. In a new field, the positive trials tend to see the light of day earlier. The negative ones are less newsworthy and may take longer to come out. And also, authors may be less enthusiastic about putting them out when they haven't got a positive result. And then the final point, as I've just demonstrated, is effect size tend to drift down as more information accrues. Next one. So what's the difference between a systematic review and a meta-analysis? Well, a systematic review is the whole sort of thing put together. The Cochrane Library one on ivermectin, which we're going to look at just now, essentially is a pre-specified plan, a search strategy, selection strategy, rules-based study inclusion, and a whole lot of issues that are reported beforehand and stuck to in terms of how to analyze and report your thing. And you end up with a document like this Cochrane review, which is well over 100 pages of incredibly hard work by a group of people, a team. A meta-analysis is really just a software technique. I mean, it basically is the thing where you bung all the trials together, and you can do this literally within 2 or 3 minutes. So you can generate those forest plots like the one shown at the bottom in 2 or 3 minutes. That's how quickly the software can give you results. And if you put apples and oranges together, you end up with a fruit salad. So you've got to be very, very careful that what you put together actually belongs together, or you may be at high risk of making false deductions from those. So meta-analysis is a subset of the stuff you do with a systematic review. Some systematic reviews say that they really can't put out a meta-analysis or a forest plot because the information is so weak. Let's carry on. So here's the famous one from quite a long time ago now, and I've put it up, and I apologize for the quality of the slides, but that's because it is actually from quite a long time ago. It was published in the BMJ in 1995, and the title says it all: Misleading Meta-Analysis, Lessons from an Effective and Simple Intervention That Wasn't. And that was talking about magnesium in people who'd had a heart attack. And essentially, if you look at that little graph on the left of the two graphs, the one on the left shows that there was a whole bunch of studies scattered together having positive effects. And from that, a meta-analysis was done in about 1992, recommending that magnesium was good stuff for people who'd had a heart attack. And the total number of patients who were then available at the time of that was about 1,300, with 78 deaths. The ISIS-4 then came out, and I can't remember off the top of my head, but I think it was about 57,000 patients, and showed unequivocally that magnesium did absolutely nothing. And it's important to remember this, because just because you've got a meta-analysis doesn't mean you've got truth. It can still change. And yeah, all right, let's move on. So I want to now move on to another thing which is fraught, which is this business we just touched on, and that is about what happens if your comparison group, in other words, what should be the placebo, actually has a medicine in it that might have pharmaceutically active properties as well. And the example that's fairly topical at the moment is the combination of chloroquine and azithromycin. COVID itself tends to lead to QT prolongation. Azithromycin does too, and chloroquine does as well. So if you put all three together, well, firstly, the top forest plot shows that when chloroquine on its own was looked at, it didn't seem to do either benefit or harm. But when you added the two together, and again, this is very weak evidence, only small numbers of patients in a limited number of trials, there was a suggestion that it might do harm. So if you have a study protocol, for instance, that suggests that you give standard of care for COVID to all your patients, and some of the earlier studies regarded azithromycin as standard of care, although we now know it does nothing for COVID, then if you had your control arm having chloroquine and your active arm being ivermectin, you would effectively be comparing a QT sparing agent, which would be ivermectin, against two agents which prolong QTs. So the active comparator is potentially problematic. It introduces risk in how we perceive a perceived benefit from ivermectin. It might have been in a study like that you just simply weren't using chloroquine plus azithromycin together. Next one. And then this final pit stop is, so meta-analyses are only as good as what you put in, and generally speaking, they're very vulnerable to change if all the trials have small numbers of patients. Combining studies which don't really belong together will increase power, but it doesn't necessarily bring greater truth. Next one. All right, and then this is just a little sort of detour into the issue of where we in terms of what we do. So Trudy's already mentioned the issue of assessing evidence, which is benefit and harm. Then from that, you end up with guidelines, which are available. And from that, you get recommendations of what to do. And the problem with this is that quite a long way down there, you really should still be having recommendations that are weak and need high quality data before they can produce a strong recommendation. Next one. Right, let's get on to the background of ivermectin. I'm not going to take you through how it might work and all that, because that stuff has been in the public domain for a long time. We all know about the 100-fold factor in terms of the difference between what was used in vitro and what actually can be achieved in a human body. But be that as may, here's the stuff that it really does work for. On the left, you've got Sarcoptes scabia, and it helps a lot for crusted or Norwegian scabies. And on the right, I didn't want to, this close to Stappa, put in a picture of lower in real life, but that's a Giemsa stain of a worm that it also works against. Next one. And this is the indication for which it's registered in South Africa, which is rosacea. It's registered as a cream, and because the cream is registered as such, the chemical entity in the cream is also now registered in South Africa. There's no pill or capsule that's had a quality control approval tick from SAPRA, and I'm sure you're all aware of those studies that study that looked at the other medicines that were sometimes found in the medications, the pills that were floating around in South Africa. But anyway, so the opportunities for its use for indications other than the registered indication of rosacea is a Section 21 compassionate use program that's been in existence since January and is a fairly straightforward but paper-intensive process. And then there is the issue of writing a script for an individual patient and sending that to a compounding pharmacy, which technically then makes up the product for that patient, and it has to then be used quite promptly and for that patient only. It's off-label use, so it requires informed consent. You shouldn't really be advertising it. Patients should be aware of the hazards of compounding, particularly if unlawfully sourced products end up in the mix, and there should be a record of discussion on efficacy and safety issues. And if you want more detail on that, Medical Protection Society has that link will go to there, and they've covered it in some detail. Trudy, the next one. So the duty of care, this is also from the MPS website, is that discussions relating to the use of ivermectin should be based on the most recent clinical trial data available, and the limitations of such data and the consequent limitation in terms of ability to fully advise patients needs to be expressed to patients. Now let's just look at the data, having got that in the background. Next. So the first thing is that there are a whole bunch of endpoints we could look at. Just in terms of brevity and to make sure we stay roughly within our timeframes, I'm just going to talk about mortality, and we can go on to the next one. This is our review. Trudy's already told you what it says. It's essentially saying we really don't have enough high quality evidence to recommend its use other than in the space of a controlled trial. Next one. So Trudy's already mentioned Al-Ghazal. Those of you who are sort of reading the ivermectin literature may be aware that this study was recently withdrawn, and the reasons were given that there were some problems with the actual data itself and possibly with the way it was written up in terms of it not being original text. But be that as it may, it's off the table at the moment. As far as we're concerned, we put it off the table long ago because it had an active comparator. So it's not really been something we were particularly excited about. So it hasn't been something that's in either our equation or that of the Cochrane. Next one. This is another trial just to show how you have to go into these things in a fair amount of detail before you realize what's actually going on. This is a study from also the Middle East by Nayi and co. And the reason the Cochrane rejected it was because 46% of the controls were PCR negative. Okay, so we weren't, most of the trials they wanted to look at were all PCR positive patients with COVID. The other problem there is that there was a difference between the control and the treatment group. 46% versus 20% is a massive difference in a so-called randomized trial. And it's really so imbalanced that it's difficult to understand how that happened. The other fun thing for those who like looking at the detail is if you look at diastolic blood pressure, there's a median and an interquartile range, and all three numbers are 80, which means that exactly half of the patients in the study had a diastolic blood pressure of 80, not 79, not 81, but 80. And clearly that was something that needed to be cleaned up. And that's why this is still a preprint. It hasn't actually been published yet. Next one. All right, so what does the Cochrane review show? What they could find that they were prepared to include was for moderate disease, for mortality. They found 2 studies and had a grand total of 14 deaths in that group. They were split like that, and there was no message to suggest that there there's any difference in outcome between those two groups in terms of the confidence interval around that effect size. The next one, the same applied, sorry, Trudy, next, there we go. The same applied to the comparison in milder disease, and there they only had 2 trials again. One trial didn't contribute any patients because nobody died in it. It was tiny, with only 12 patients in each group. The big one is Lopez Medina, and that had a larger number of patients, but only 1 death. And again, very, very low quality evidence on which to base anything. Okay, if you just keep those 2 numbers in mind, there's a 200 and 198 over there, and that's quite interesting because we'll come back to that. Next one. Right, so this is a study by Bryant and co. His one of his co-authors was a South African, but we'll just come into there. I've just put it up to explain how these things differ. So we've just seen that the Cochrane group had almost no trials left. This has a fair number of trials. If you look, there's a lot of names printed on the left, but there's a lot of noughts in the control in terms of events, okay? So it goes 0, 0, 0 for many of these things. In the mild COVID, that top section on the left, there's only 1 event again. In the control group, there were 12 deaths. We have to take out 4 because Elgazar contributed those. So we left then with 8 deaths versus 1 on which to base a decision about efficacy in mild disease. And the same sort of thing happens with more severe disease, the middle block there. And Nair has been separated out. He's sitting there at the bottom and has his own little group because they were mild, moderate, and severe. The other thing to look at is on the right is a risk of bias table. And as you can see, some of the little red block, which means that there was considerable concerns about whether the trials could be assumed to be bias-free in terms of how they were conducted or all the other features that we talked about right at the beginning. Next one. So this is just looking at it in more detail. Again, I just want you to focus on Lopez Medina in the middle there, and you see this figure has changed. It was 200 in the Cochrane review, it's 275 here. Now, I don't want to make a fuss about that, but I just want to demonstrate how it's quite easy and quite normal for these things to change between one meta-analysis and another. And it's these sort of small changes which can lead to changes in confidence intervals around effect size. Next one. So then we're left with more severe disease. Again, Elgazar is contributing quite a lot of the evidence there, but already there wasn't evidence of a effect in that subgroup, although overall there was still an effect. Let's see the next one. So what I've done here is I've just done basically a sensitivity analysis, which is where I've taken the overall effect size for mild and severe disease and taken that top one is from the Bryant study, exactly the same number. And then they already had taken out high risk of bias things. So the second bar down is also theirs, still showing apparent benefit. When you take out Elgazar and another small study with an active comparator, you suddenly no longer have a significant benefit of the agent. I also have taken out Nair for the reasons which I've explained, that, you know, in other words, there were some issues around that in terms of balance and whether the patients actually had COVID or not. And if you add back a new study from Argentina, you show that it's now sitting almost completely on null. The Cochrane one is the one below that. The Cochrane one and the one without Nair are almost identical, 0.65 and 0.67, with no evidence that this is a significant difference. And the bottom block there is what happens if you add the new Argentinian study to the Cochrane review. So you see that you have to try quite hard to find a review or a endpoint for mortality overall that is significant. And that's because the evidence is just so weak. Next one. So finally, just to end off then, you know, for me, this has been a very interesting journey because I'm fascinated by the difference between what we were thinking was an appropriate comment on ivermectin and the way it's being used in the community. And one recognizes that society expects you to balance scientific integrity as a doctor and empathic care. And we have got a society that says, look, listen to these bodies. There's professional bodies which guide practices, and also HPCSA, SAPRA, and there are also medical legal checks on this. So here we have a situation where most of the guidelines up until now that have appeared have said, don't use this agent. We don't know yet whether it works. And yet there seems to be an enthusiasm for prescribing it based on a less scrupulous or careful review of the evidence. Next one. So whose truth is it? I mean, who's right here? Next one. I think one of the things that I was thinking might make a difference here is that it was an empty space. COVID is a horrible illness, and we really didn't have and still don't have magic medicines that are going to make much of a difference. If somebody came in and said this thing makes a difference, it's a very appealing, positive message. And people have not been enthusiastic about looking at the weakness of the data. If people in a group which you respect are using it, you may also be enthusiastic about joining that group. We have a human beings this desire to take pills for centuries, that anything you take gives you a feeling of agency over your illness. And there's also this issue that the messaging that's come out doesn't have consequences. You can say it, but it doesn't seem to have led to any deleterious effects. Last one, I think. Next slide. So are we there yet? I mean, I think that what we can say is that the probability of any benefits is at the moment in a reduction phase. It seems to be becoming less clear. The issue around using it because it can't do any harm has got a number of problems with it. People are now seeing it as a vaccine substitute, which is clearly very problematic. There's people who say, well, let me just take the ivermectin that somebody prescribed for me. I'll wait a day for it to work before actually going to hospital. If we go around sort of pushing medicines which really don't have enough evidence, there maybe are threats to our professional integrity. And there are also legal impacts which may arise down the line. I'm concerned that this, because people are reading the top level of the data and not plunging down, we may be losing our ability to detect things that really aren't quite as clear as they should be. And are prescribers really aware of the evidence and providing informed consent? So I think I'll stop there. I'm sorry I went a little bit over time, but I think that it's hopefully got you some things to discuss now. So great, there we go.
Thank you very much, Andy. Thank you so much for a really nice overview of the state of evidence in terms of ivermectin, and most importantly, the processes behind it and why the current guidance from the NEMLC subcommittee says what it does, of course, which is concordant with the guidelines from WHO and other international organizations. I am aware that it seems many people seem to have difficulty with the chat today, and I think teams are struggling with the large numbers. So what we're going to do, please, if you do have questions now, do type it in the chat. If for whatever reason you can't type it in the chat because some people seem to have difficulty with that at the moment or access to it, then what I suggest is stick up your hand and we will try and unmute you and you can ask your question verbally. So let's perhaps go from there. And while I've been getting messages on email, I've getting messages on WhatsApp, I've been getting a whole bunch of questions that have been coming through as well. So while we wait for that, perhaps Andy, the first question we could ask is from a colleague who says that is it not the responsibility
Okay, Jeremy seems to have got stuck a little bit at the moment. If there are any questions that people want to put their hands up for, let's do that now. So I'm seeing some hand. Well, there's. Okay, I think, yeah, I don't know where to start because there's hands, but there's no. All right.
I can help unmute Andy if you call a name.
I'm not seeing the names at the moment, though. That's my problem also. So if you can show names, then we can just go for it.
Sorry, it seems to be that there is some chat questions. Can you see the chat, Andy?
Yeah, I can see. There's one about dosages. I can see that one there. And I can just address that maybe. That's from Lucille Bloomberg. You know, the issue she's saying is how do you evaluate dosage and safety for a drug where the evidence is really for other indications? Now, I mean, I think that that's always the problem when you have a new medication or new indication for an older medication. You have to start almost from scratch in terms of working out whether it will or will not work. And one of the issues then is building on previous studies' doses and seeing if that can be tweaked. The bottom line of it is I think that there's just so many doses. Every single study seems to use a slightly different dose and a slightly different duration. So at the moment, it's all over the show as far as I'm concerned. I honestly don't know how anybody is knowing which dose to use. They seem to be using doses based on what was out there and we used initially, and those were based on those earlier studies, which, as we can see, some have been debunked and others are, I think, relying simply on doses that the prescribers were familiar with from its use in other conditions. So the short answer is I don't know, and I'm not actually that interested in dosing until such time as we've got stronger evidence of efficacy.
Andy, I'll try again. Sorry.
Okay, welcome back.
Not being my friend today. I'm going to keep my video off. One question that did come through to me as well while we were talking was about whether there's an obligation on the part of those who use drugs like ivermectin off-label, so to speak, to add to the body of evidence. And what would you say on that perspective? Because we're obviously getting a lot of off-label use, but we have no real evidence coming through at all despite quite a lot of use in South Africa.
Yeah, I mean, I think that that's fascinating. As you know, we were just discussing earlier the trial from Argentina where to get people, because so many people are already on ivermectin, that it was difficult to get people to join a trial. And it really has to do with how society is seeing this agent. You know, I mean, we have said as a committee right from the beginning and continue to say that people who are taking it should be part of controlled trials. And arguably, when you prescribe it off-label, you aren't adding to that body of evidence. And in fact, the same argument applies to small, poorly organized or poorly conducted trials too, that they actually don't add much either. You know, you need actually quite strong evidence before things become clearer. And so even if you do take part in a tiny trial with 30 or 80 or 50 patients, you still won't be much closer to an answer.
Yeah, it's a great question. I mean, one of the issues which Professor Bloomberg's just highlighted as well in the chat was about, you know, this translation. I mean, ivermectin is usually only given once or twice for the current indications for which it is recommended. You know, and this is, you know, it's a big extrapolation potentially to give such high doses for such a long time, and the potential for adverse events on that. And I think that is, as you say, it's a huge challenge. And then one of the other questions which came in was really about perhaps prescriber pressure. So saying, you know, if not ivermectin, what can be given to people who want a quote antiviral when you visit your general practitioner, or where you come to a specialist to be seen. And I think the prescriber pressure is perhaps part of what drives this in this particular sense. I don't know what your thoughts were on that.
Yeah, I mean, I think it's a fascinating thing, and from a pharmacoepi perspective, I've been looking at prescriber pressure for decades. And if you look across Europe and antibiotic prescribing, there's a massive north-south divide in terms of how much antibiotics get used for certain conditions. I was chatting to a GP once from Holland or the Netherlands, who actually said that if in her practice she gave an antibiotic to a child with a otitis media, the whole practice would be ostracized. And it's really, this is where we're sitting with our society in South Africa. We have generated as professionals a expected need. People expect, it's almost an anthropological thing, to get a prescription filled when they visit a doctor with a viral condition for which there is no current working antiviral. So it is an interesting space, and I'm well aware of the pressures people are under. But the issue is then, where does the professionalism come in too? Do we not have a responsibility to actually say, look, there isn't anything? Every time we actually pretend that there is something, and that's really where I'd go. I mean, arguably, every time you give somebody an ivermectin, you are actually saying, I think it works. And unless your counselling has been really strong and saying, look, I really don't think this stuff works, or I don't think there's evidence that it works, we're becoming part of the problem. And I think that is where it's gone. But I think social media has a massive role to play in this too. It's swept across the country as the thing to do. And it's all based on anecdotal evidence. Somebody got better when they took it and had COVID. And the problem is that, 99% of people get better when they have COVID anyway. So that sort of evidence really doesn't work, but it's very real for individuals.
Yeah, and I particularly appreciated your point as well in your presentation about the potential harms, because I think one of the lines that's often used is, what's the harm in this drug? It's a quote safe drug. And I always find that a little bit odd, because the quality of evidence for safety is of the same quality as for efficacy, namely not very good, at least when used for COVID-19. But I think many of the people who may use it or prescribe it are often sort of taken in by this argument of what's the harm. But I think you covered that really nicely in your own talk about the potential harms. We've certainly seen some of them anecdotally ourselves, although we should try and stay away from anecdotes in general. One of the questions which keeps coming up as well, both in the chat and outside, is this perception that the vaccines were registered with much less problem through SAHPRA and throughout the world, whereas ivermectin hasn't met the same approval, and saying that the vaccines are still in clinical trials in some senses, and they're much less known to be safe than ivermectin. So I clearly I don't agree with that sentiment, but I wondered what your thoughts were on that in terms of specifically this perceived disconnect between how the vaccines are treated and how ivermectin is treated.
I think it's like all these things are, an order of magnitude thing. I mean, we've been looking at some of the vaccines recently, and it's just astounding if you think of the numbers involved. So for vaccine efficacy, even a small data set would have, several thousand, 10,000 plus patients in the for whom there are recorded outcomes. Okay, and that's the sort of frames you need to be able to say something about safety. Okay, most efficacy endpoints might be reached with smaller numbers, particularly if you're looking at very severe patients, and also if they had. But once it comes to rarer things, you really need to accrue lots of patients, and it's just on a different. It literally is an order of magnitude wrong for ivermectin. We just don't know anything enough about the number of about the side effect profile, because you need a whole lot more patients. So vaccines are new, and the data is accrued very fast. But in terms of the size of the data sets, it's similar to what it's been for previous vaccine trials, and that's what FDA registration requires. So, it's gone very fast, and there's been overlapping of earlier and later trials on vaccines. So they moved really well, but that doesn't mean it wasn't done properly.
Yes, and I think the other added complication is the FDA still has the vaccines under emergency use authorization, which is leads sort of linguistic ambiguity to their status, if nothing else. But as you say, the trials there were generally much bigger and much better conducted, to be frank, as well. Tens of thousands more people than in any of the particular ivermectin trials. Just looking through the chat for some of the other questions. One issue that comes up sometimes is and it's really an evidence-based medicine question, is using the trajectory of COVID in certain countries to say that a drug like ivermectin works. So, for example, you'd look at a country saying that ivermectin was started being approved in October, and in November the caseload declined. Now, that's a sort of superficially persuasive a line of argument, but there's clearly issues with it, an ecological fallacy and others. I don't know if you wanted to maybe address that and the kind of perils of using that very, very high level data as anything persuasive.
Yeah, I mean, I think the simplest example of that is what's called the Texas shot, where you take aim at the barn door with your shooter, and once you've finished shooting, you then march across and paint a target on, and showing what a wonderful targets person you are. So this is the trouble with those things, is that they are all post hoc analyses of observational data, and it is very problematic to tease out what is cause and effect there. So in most biological systems where there's observational studies conducted, there's a thing called R-squared, which is a measure of how much of the variance of the variability in your data set is explained by the various things that you put in. And those numbers are astoundingly low usually. They explain half of it most of the time if you're lucky, and sometimes less. And these sort of studies are no exception. So there's so many factors that come into account. And, literally, if you were to want to design one of these things, it's the obvious thing is, as epidemics get out of control and surges happen, people scratch around desperately, as we have in South Africa, for anything that might help. And so ivermectin usage will rise as the epidemic goes worse, and then it peaks by itself, and usage will then drop. And that's two things going up together. There's nothing to suggest the two are in any way causally related. They're associations, not causations.
Thank you very much. So I think we are over time, but I did want to continue because we wanted to take as many of the questions as possible. Perhaps we just take one last question. It was in the chat, and so it's about the sort of bidirectional problem with people who take ivermectin. So in one case, do you as a healthcare practitioner have the right to refuse if a family member almost demands ivermectin from you, or says, if you don't prescribe it to me, I'm going to sue, or I'm going elsewhere? And that has happened, at least on the fringes in South Africa at the moment. And conversely, sort of related but separate problem is, do hospitals have the right to say to a patient, you are not allowed to take your own ivermectin while you're under my care in say, an ICU or a high care environment?
Yeah, the sort of thing you end off with is a half hour's ethics discussion. No, I mean, I think you can, chip in on this one as well. I mean, my concept of this is that really it's about whether you as a practitioner are prescribing it as an active act, or whether they're already on it and just continuing it. If the sort of event has already happened, there's no evidence that they're getting harm from it, they're sort of squashing it and stopping it it may be seen as a more aggressive act by families and relatives who really, really looking for something to help. But, to actually prescribe a medication for which there is such weak evidence, and where almost every sort of body that is giving us advice on this is saying don't do it outside of a trial, is a perfectly reasonable and medico-legally defensible position to be in. I understand that in the States, people have sued for their right to take ivermectin. It's absolutely true. It's their right to take it, but that doesn't mean that you have a duty to prescribe it. And in South Africa, where there isn't a product that is necessarily there, I don't think you have to push and or be pushed into prescribing it. But I do recognize that there's an enormous amount of pressure on people.
Yeah, thank you. And I see there's a question always in caps lock, which is always a sign of distress. So if 99% of people get better anyway, why the need for a separate-proof vaccine? Well, presumably for the 1% who don't, and for the many who will suffer plenty of adverse consequences from the virus along the way to getting better. We've also got long COVID and other things to worry about. So I think we can end it there. And thank you very, very, very much. I'm gonna try not to switch on my screen at the moment, because I think that seems to be a trigger for Zoom to get me out. But I just wanted to go through a few thanks very briefly. Thank you very much to MRC and Cochrane South Africa for hosting this series of webinars on COVID-19, and especially to Tamara Crader and to Joy Oliver, who are really the powerhouse behind this. Thanks also to the Better Health Program and the NEMLC subcommittee on COVID-19 therapeutics in particular, and to today's speakers, Trudy Lin and Professor Andy Parrish. So thank you very much for dealing with this hot topic. Many people have asked how to get a hold of a recording of this. There is a link which we provided in the beginning. What I'm going to do is try and share my screen with that page on so that you can see how to get hold of it. And they will be uploaded shortly, I'm sure. But at this point, what I'm going to do is thank everyone for a great session. And I'm going to, there we are. So hopefully that will come up there. You can see how to access them. But we're going to close the meeting at this point. Thank you all for your attention and for your patience as we work through a few of the technical glitches. Will hopefully have resolved by the next time. The next meeting is on the 24th of August. We'll be covering vitamin C, mucolytics, and a few of the other aspects to this. And we hope to see you there. Thank you very much. And I'm going to just leave this slide on in case anyone wants to take the website address down. Thank you very much.